Good morning. This is Shan from the University of Massachusetts Medical School and the co-chair of today's symposium. On behalf of ISV, its board and officers, welcome to everyone. After successfully running the COVID-19 Vaccine Congress series in this past summer, ISV is very pleased to organize this North America influenza vaccine update. Influenza remains a major threat to global health, and the goal of a universal influenza vaccine is far from being accomplished. ISV would like to thank Center for Vaccine and Immunology at the University of Georgia to co-organize this symposium with us. We would also want to thank the speakers and the chairs as the leading expert in this area to share their most updated progress in influenza vaccine research. I have no doubt our audience will enjoy today's symposium. Now let me introduce Professor Ted Ross, current president of ISV and the symposium chair, to start today's official program. Thank you.
Thank you, Shan. I appreciate the introduction. As you said, I am the president of the International Society for Vaccines for 2020 and 2021. This has been a challenging year for ISV with the COVID shutdown, but we've actually been able to grow our organization over this past year, and the outreach through these symposias have been extraordinarily successful. And so this one that we're going to do today is focused on a very important program that has been funded by the National Institutes of Health for the CIVIC program to develop universal influenza vaccines and test those in both preclinical and clinical settings. And so several speakers today, all the speakers today, are associated with this program. And so I'm going to introduce the first two speakers. Dr. Georgiou from the University of Texas. He is an endowed chair, a professor at the University of Texas in the Department of Chemical Engineering, Molecular Biosciences, Oncology. And so he's going to tell us today about his studies on molecular deconvolution of the serological antibody repertoire following influenza vaccination. And now I turn the meeting over to Dr. Georgiou.
Thank you, Ted. Thank you, Shan, for inviting me to this meeting. The program seems really impressive, and I am grateful for the opportunity to participate. I would like to spend the next 20 minutes or so to discuss some of our work on the molecular composition of the serological response to influenza. And as I'm sure you all know, we have enormous amount of information on titers, binding and neutralization titers, and also on individual B cell encoded antibody from peripheral B cells, whether these are antibody secreting cells, plasmablasts, or memory B cells. However, ultimately, the response and the serological memory comprises of a multitude of antibodies, and understanding what the nature of these antibodies is and being able to infer and learn from that information lessons that might be relevant to how protection occurs, the longevity of the responses, and other issues that we will touch on later on is significant. So can I have the next slide, please? So this is the outline of my talk, effectively talking about discussing some of our findings on the serological repertoire. It's a relatively recent field of inquiry for us. We've been working in this field in influenza in particular for the last 5-6 years. But one thing to keep in mind is that when we talk about the serological response, the clonality of the serological response, the number of antibodies that comprise the circulating antibody pool, is substantially lower than the diversity of antibodies that can be found in B memory cells or plasmablasts at day 7. And there are a number of reasons for that we can discuss later on. But overall, our studies with multiple vaccines have demonstrated that the clonality of the antibody response is at least 3 or 4 orders of magnitude smaller than what we see from B-cell cloning. The objective of what we're interested in doing is understanding how diverse is the response, how many different monoclonal antibodies comprise the serum response, and of course, find their sequence, the relative amount, and also what they bind to and how they may contribute on their own or as a group to protection to confer neutralization via different mechanisms, and I'll discuss some of that later on. The temporal dynamics, especially for the serological response, matters a lot. How many antibody clones in circulation persist for a long time versus antibodies that may be lost after a few months. In other words, they may be transient. And ultimately, that leads to the fundamental question of what properties of an activated B cell eventually are responsible for expansion of that particular B cell to have a number of clonal progeny and differentiate to long-lived plasma cells such that it can contribute a significant amount of antibody for the serological response. We have been working on the clonal relationship between the IgG and the IgA repertoire. It's actually quite interesting. Understanding how the clonality and the features of serological repertoire change with time, and a number of other issues. Can I have the next slide, please? For that work, it was imperative, in fact, to develop a technology that enables the analysis of the antibodies in serum. The technology, the pipeline, which is fairly complex, relies on LC-MS/MS proteomics of serum antibodies that bind to antigen and are enriched by affinity chromatography on immobilized antigen columns, but also information on the repertoire of B cells, especially B cells in which we link the heavy and the light chain in one amplicon and sequence both the heavy and the light chain simultaneously, so we can get the native paired antibodies that are found in our donors. Suffice to say that from that information, we obtain clonotypic information. In other words, in more technical terms, we spectrotype the CDRH3s of the constituent antibodies in serum, and we can determine their abundance from the liquid chromatography portion of the LC-MS/MS. And after that, we make the antibodies recombinantly and study their characteristics, how they bind, and in several instances, move on with structures. Next slide, please. So let's dig in directly into influenza. I'll briefly describe some of our findings from an earlier study published 4 years ago in Nature Medicine. We were fortunate enough that the story made the cover of that issue, where we first reported on the serological repertoire of a small cohort of young adults. I should mention that these studies are actually quite time-consuming. The analysis of the serological repertoire, because it involves, as I mentioned before, both relatively elaborate B-cell sequencing and quite long LC-MS/MS runs and heavy informatics, and therefore it's not really very easy to analyze large cohorts. So all our cohorts are typically less than 10 donors. But to make a long story short, one thing that was apparent in this study is that when you look at the repertoire to flu zone, regardless of the strain, and you look at the serological memory 6 months after vaccination, a large number of antibodies, in fact, the majority of the antibodies, could be found in the serum before vaccination. So effectively, there is a very strong boost effect, which one might expect. There were also, of course, some vaccine-elicited or new clonotypes. These constitute either antibodies that were present at very low levels at day 0, therefore could not be detected by LC-MS, or de novo clones elicited by the vaccine. Looking at the cohort on the lower right side of this diagram, about 60 to 70% of the response is effectively boosted antibodies that pre-existed. And this is something that's important to keep in mind. This is actually a recurrent theme in most of what I will discuss later on, and not totally expected since everyone here is quite sophisticated and knowledgeable about flu. May I have the next slide, please? To elaborate further on the issue of persistence of certain antibody clones and how the serological memory over longer periods of times is impacted by the initial exposure effects, we looked at a unique individual that has been studied very extensively by Lanzavecchia and Corti, and from whom many very potent neutralizing antibodies binding to the stem had been isolated. Unfortunately, Antonio and his team had serum and blood from a number of years. The donor had been vaccinated multiple times, primarily with California 09. And we looked at the repertoire to California 09 over time. And what you can see over a period of 5.5 years, and this is, by the way, a typical example of how we present the data in these experiments in the form of a histogram, whereby the y-axis represents the relative abundance of a clonotype. We have validated that the abundance is, semi-quantitatively accurate by labeling, by using labeled peptides, by the way. So each of the columns represents a different antibody clonotype in serum and its relative abundance. And as you can tell, the clonotypic diversity of the repertoire in this donor is typically about roughly 25-30 antibodies, of whom a large number, especially a large number of the antibodies that are persistent in circulation, continue to persist over years and years, and they persist both in the steady state response as well as in the response immediately after vaccination. In other words, when the titer is higher, is at its highest about roughly 28 days after vaccination. And this is shown on the lower right-hand side of the plot, where we show the composite data for all the donors. So again, about 70% or more of the response seems to be invariant. Now, granted, this is in a donor that was immunized with the same vaccine component, the same H1, but nonetheless, it is quite, I think, important to keep in mind how many of the antibodies in the vaccine response are boosted pre-existing clones. Can I have the next slide, please? One of the interesting things that came out of the serological studies was from the analysis of some, from the analysis of some of the top most abundant antibodies, especially in the young donor cohort that I mentioned earlier. And when we looked at some of these antibodies, we wish to basically express those proteins. Let's look at the top line, D1, H1, 3, H33. This particular antibody was the third most abundant in the H1 repertoire and the third most abundant in the H3 repertoire. In other words, the antibody bound to both H1 and H3. When we looked at binding across both group 1 and group 2 strains, we saw incredibly broad binding, and that was the case for a number of other antibodies that bound to both H1 and H3, which is rather unusual. Now, ordinarily, one would speculate that these antibodies may be directed to the stem region of HA, since this is more conserved. However, that was not the case, as I will mention later on. None of these antibodies neutralized. They had no HAI titer or neutralized in microneutralization assays. Nonetheless, Florian Kramer and his group were quite interested in testing these antibodies in mice, and lo and behold, actually the antibodies conferred very good protection either prophylactically or when administered after infection in a therapeutic setting. So in other words, these are non-neutralizing antibodies that are protective, that are very abundant in serum, and for which the protection is solely due to FC functions as opposed to the usual correlates of protection that we normally expect in influenza. Well, okay, this may be an observation that's somewhat interesting, but out there, however, as it turned out in the following years, a number of other groups, namely Jim Crow and Steve Harrison, among others, can you please move to the next slide? It turns out identified a large number of other antibodies that actually bind to the same epitope. And that epitope is a highly conserved region in the trimer interface. We had reported that initially, and that was actually verified and expanded upon by a number of different antibodies that more or less, in many instances, display a highly focused way of binding to the antigen. So the net conclusion, the overall conclusion here is that a number of donors contain in their plasma antibodies to a very conserved head region in the trimer interface, and those antibodies actually do contribute to protection, and they contribute to protection via an, let's say, non-classical mechanism, in other words, via FC functions rather than neutralization, which is what is typically, as I mentioned before, looked at. Can I have the next slide? I'd like to wrap up and discuss some more recent work that has not yet been published on the impact of age on antibody responded to Fluzone. We looked at the response to the vaccine. We looked at the repertoire to intact immobilized vaccine components, egg-based inactivated vaccine that are used in the Fluzone vaccine in a cohort of young, middle-aged, and elderly donors. This is a cohort that came from the University of Georgia and a collaboration with TED, as well as Sanofi Pasteur vaccines. And one of the things that was very abundant and very noticeable from the beginning of the study is that when you look at the repertoire to the vaccine in younger donors, a small percentage of the antibodies are cross-reactive between the H1 and H3 vaccine component. That was, by the way, also true in our separate cohort that I mentioned earlier. And most of the antibodies are specific to that particular vaccine component. However, this is not the case for the middle-aged and the elderly group. Next slide, please. This, in fact, shows the distribution of antibodies with respect to whether they were monoreactive, in other words, bound only to one vaccine component, or to both as a function of age, where the green portion of the pie plot constitutes or refers to the cross-reactive antibodies that bind both to VH1 and VH3. So, especially in the group to the right, the over 65 group, it's quite remarkable because the repertoire, the response to the vaccine is overwhelmingly composed of antibodies that are actually cross-reactive. And in some instances, they constitute 90% of the serum response. This is shown on the panels below. And also, I want to point out, for those of you who are interested in these features, that the antibodies that are cross-reactive are more somatically hypermutated. Next slide, please. Now, when we look at the anti-HA antibodies, those that are specific to a particular vaccine strain, they're not cross-reactive. With age, we observe that these antibodies become broader. In other words, people, as they age, have anti-HA antibodies in the serum that have greater breadth, binding breadth, than people that are younger. And this is consistent with a number of studies by Patrick Wilson and several others, Bill Robinson and etc., that have analyzed the B cell, the peripheral B cell repertoire. We see the same thing in the plasma. You get broader breadth, which is also consistent with serological data. Can I have the next slide, please? Now, the interesting thing is to understand what are these antibodies that bind to both the VH1 and VH2 component. And as it turns out, what we saw here is again a pie chart where we plot the different antibodies and their specificities for those antibodies that we could make and characterize for 3 representative donors. And usually those antibodies cover about roughly 60 to almost 80% of the total response. Now, what you see in the green antibody pie piece is that those antibodies bind to essentially conserved portions of the flu vaccine. In other words, they are not directed against HA. The broad antibodies that bind to the trimer seem to be predominantly enriched in the young, but not so much, they seem to die off, in other words, not be as represented in older groups. And instead, the antibodies that bind to both components of the vaccine, bind to a nucleoprotein, bind to M1, M2, and so on the virus components that do not change more or less from year to year. If you think about it, this is not unexpected. Every time you get vaccinated, you get more and more antigen, and the immune response becomes perhaps increasingly focused to the conserved portion of what it is challenged with. So in older adults, overwhelmingly the antibodies in the serum response seem to target conserved influenza antigens. May I have the next slide, please? One particularly interesting and puzzling group of antibodies that we saw in these donors turn out to bind to glycans, and these are avian glycans that are very rare in humans. These glycans, these antibodies to glycans bind to the vaccine, they bind to denatured vaccine. However, they do not bind to recombinant protein, which is not, of course, produced in eggs, but they do bind to egg allantoic fluid, which is highly enriched, of course, in egg glycans. Using glycan arrays, we identified the type of glycans that's recognized by these antibodies. These antibodies constitute public clonotypes. They are seen in different donors, and they're more or less originating from similar lineages and have certain stereotypical features in the CDRH1 and CDRH2. And they're not overly abundant, but they can constitute about 10–15% of the repertoire. So from the point of view of protection to flu, likely they do not contribute much, if anything, since they are directed against an egg antigen, which is not present, obviously, in influenza. Now, I should point out that in MDCK cells, you can also find similar glycans and response to some of the glycans in the inactivated vaccine, which raises the question, well, what do you do in terms of vaccination strategies? The recombinant subunit vaccine, of course, does not have these other extraneous components, most likely, although you never know because you haven't looked at anti-insect cell antibodies in flu block donors. But of course, it's lacking the other components of the vaccine, which have been established previously to be responsible for strong T-cell responses, like NP, for example. And also there are some indications with M2E specifically that some antibodies to these components may be protective. Of course, also antibodies that are protective and are eliciting a lot of interest right now to NA. So to make a long story short, the net result is that, or the conclusion I'd like to leave you with is that as you go older, it seems after repeated vaccination, you get an enrichment in the response to egg-based vaccine of significant portions of antibodies that bind to non-HA or NA epitopes. This can be a dominant part of the repertoire, and several of these antibodies bind to egg glycans. So this is just a snapshot of the kind of things that, the kind of analysis and findings one can derive from looking at the serological repertoire. We're currently looking at in more detail in collaboration with TED on the IgG and the IgA repertoire, and also we're very interested in other features of the serological response, especially polyspecificity and of the antibodies that are elicited by vaccination, which I think will become a significant issue or an interesting issue in vaccine development in the future. I'd like to thank you for your attention and thank my co-workers. Can I have the next slide, please? All this work was done with Greg Ippolito. Many of you know Greg. He's a B-cell immunologist. We have been collaborating for many years at UT Austin. And Ji-Won Lee, he has now set up his own lab at Dartmouth. Several other very talented students and postdoctoral fellows. Ted, Tony, we collaborate and work with both civic groups, Harvard, VRC, Mount Sinai, Florian. This has been a terrific community, very collaborative, and I'm honored to be part of this community. Thank you very much.
Thank you, George. Very interesting. And yeah, I hear this is the third or fourth time I've heard you speak, and every time I learn something new about the repertoire. We do have time just for one quick question, so I'm going to read this off of the Q&A. It's from Dr. Christine Vogel at New York University. She says, for the findings of broader antibody reactivity in older participants, do you notice a change in the HAI assay? In other words, the titers, is it reflected in the level of the titers?
The levels of the titers are slightly lower, but not as low as you would expect given the response to the vaccine, given how much the response to the vaccine is skewed towards non-HA titers. So if you look at the relative quantitation, then I think the conclusion one has to come up with is that many of the antibodies that are in circulation that bind to HA are actually antibodies that are, a larger number are protective. A larger number actually confer HAA titer relative to younger donors. And I think we have some evidence. We don't have the full range of data yet to support this indication, this conclusion.
All right, well, thank you, George. We appreciate it. We're going to move on to our next speaker, which I'm thrilled to introduce, Dr. Kathleen Neuzil from the Center for Vaccine Development and Global Health at the University of Maryland. She's a professor in medicine and pediatrics. She is also one of the project directors of one of the civic clinical corps that will be running several of the vaccine candidates that come through the civic program. So Kathy, I turn it over to you, and we look forward to your talk.
So thank you, Ted. I'm certainly happy to be here. You can go to the next slide. And we heard a lot of complex immunology, and now we need to interpret how we can best measure that in terms of human performance. And it's not easy. If we could go to the next slide. So in the next 20 minutes, I'm going to give you a brief overview of human challenge studies, also known as controlled human infection models, talk about them specifically for influenza, and highlight a few of the areas where, not surprisingly, influenza makes them particularly challenging. Next slide. Just as an introduction, what I am talking about here is the intentional infection of healthy volunteers with a bacteria, with a virus, with malaria. It's been used for many, many pathogens. And then we closely monitor those volunteers to do any number of things, maybe establish pathogenicity, characterize immune responses, look at vaccine or treatments. Now, we do this because volunteer studies are more controlled and they can be performed at lower cost than pivotal trials. And I'll talk about some of the trade-offs there. And I'll even give you an example where a human challenge study has been used as the basis of FDA licensure for a vaccine. So there is a potential regulatory pathway here. Next slide. Now, this is a more detailed slide, and I'm not going to go through all of this, but again, that goes over the many, many applications of the controlled human infection model. You know, in its most basic sense, it could be used just to prove that a microbe is actually pathogenic or causes a certain disease in situations where that may not be known. You may identify virulence factors. You know, it's been used for correlates of protection, which I think is an important finding. One aspect, though, that we have to consider here is that these models are generally performed on healthy young adults. And so we really have to think about the generalizability to the populations, whether it's older adults, infants, children, people with chronic diseases, who are frequently most affected by these diseases. So that's one of the limitations of using these models. Next slide. Now, when we consider whether to use a human challenge model, we look at features of the organism and the features of the disease. It's certainly better if the natural history, the disease spectrum, and the modes of transmission is known, because of course we are trying to duplicate what occurs naturally. We would like a situation where natural infection induces immunity against disease, because that's a powerful way to validate the model. So an infection reinfection model helps tell us how good our model is. Clearly, if we're going to use this model to evaluate vaccines or therapeutics, we would like the disease to be inducible in healthy adults in high attack rates. Ideally, we want a short incubation period. We frequently do these as inpatients. We don't want to have to wait weeks, for example, for the transmission and the disease to occur. It's always good to have an available treatment for the infection. Should we misjudge the inoculum or have a participant that gets sicker than we intended? However, that's certainly not always the case. There are challenge models for dengue, for example, which doesn't have an available treatment. We would prefer to have diseases where long-term or chronic sequelae are even absent or preventable. And when we think about the organism, because these are GMP organisms that we're using, we would like to see genetic stability. We want to see consistent purity, identity, and potency when we use these challenge strains. We want to be able to identify a standard inoculum size and a way to deliver. And then we think about formulations much in the same way we think about formulations for vaccines or other biologics. You know, should it be freshly harvested? Should it be frozen? And again, generally we're looking at GMP manufacture. Next slide. And it's really important to mention the participant safety and ethics aspect of this, and we really put a lot of time into this to again be sure that we're really screening for healthy people, that we are looking at a pathogen escape or transmission, which may either be within the challenge, so it could be to our staff, our clinical staff, or we could be looking at community transmission. We have to be very careful about that as well. Obviously, we want voluntary participation. We want to be sure we're asking important scientific questions as we do with any trials in humans. And then we really have to think about public health, public trust and public health. So for many of these challenge studies, depending on the organism, we may talk to the health department, for example, if we're using an organism that does have a risk of community transmission. I show here we did a challenge study in our unit with influenza last fall. This was a Wall Street Journal article. These people got the flu, so you don't have to. This is one of our volunteers in the unit. It's really interesting to understand what motivates people to be in these studies. We had actually several authors who were looking forward to 7 to 10 days in isolation to be able to work on books and some of their writing projects. This particular volunteer actually had 5 children and multiple pets at home, and she said this was the most relaxing week and a half that she had had in some time. So different motivations for people in these trials. Next slide. Now, we do our influenza challenges in an inpatient facility, and I don't need to talk a lot about this, but there is some consideration of community risk, and of course that may depend more on what strain you're using and whether that strain is already in the community, for example. But you do have to think about strict protocols for participant discharge and plans for early discharge, and this is going to happen. It's a rule and not an exception. You know, we had somebody in their family may get critically ill. We had somebody with a legal problem, you know, and they're anxious to leave while they're still shedding virus. And so you need to think through the contingency plans, make sure the participants fully understand. Next slide. So if we look at some remarkable challenge studies that have really helped inform medicine, you know, we can look at the Edward Jenner smallpox vaccine proof of concept as a challenge study. You know, he inserted pus taken from a milkmaid with cowpox into a cut made in the arm of a local boy, James Phipps, and then several days later he exposed the boy to smallpox to show a proof of concept of protection. Next slide. Here at the CVD, we actually did a series of studies with a cholera vaccine that was developed here to achieve FDA approval on the basis of a pivotal human challenge study. And if you think of a traveler's vaccine like cholera, which was the basis for licensure of this vaccine, you're not going to have a high attack rate. It's going to be very difficult to do a field efficacy study. But you can see here with approximately, we had about 80 participants in the pivotal trial. You know, we were able to show high efficacy, and with a series of other studies, again, put this package forward to the FDA and get licensure for this vaccine. Next slide. And a more recent example that really made the news just in the last few months is, should we be looking at controlled human infection models for SARS-CoV-2? And could this be a way to accelerate vaccines? And working with others, I published this opinion piece in the New England Journal where we said, you know, we're not convinced we do need controlled human infection models for this virus, that we think large randomized controlled trials are the way to go. They're the most efficient, they're the most generalizable because they're not restricted to just healthy adults, and they're the most scientifically robust pathway to establishing vaccine efficacy. However, A controlled human infection model of SARS should be considered for second-generation vaccines to address essential questions of immunopathogenesis, perhaps duration of vaccine-enhanced immunity, or importantly, to look at correlates of protection. And ideally, we would do a SARS-CoV-2 challenge model once we have an effective therapy. At least I would feel personally more comfortable doing a challenge in that situation. And just looking at the timeline that we built for this perspective, you know, you have to consider the manufacture of these challenge strains, and that's not always easy, and that can take time. So I think, as we have shown with the Pfizer and Moderna vaccines for SARS-CoV-2, it was faster to go the traditional route to get efficacy data. Next slide. So moving now to influenza, influenza challenge studies have actually been done for decades. You know, early on, there were studies that helped to establish a correlate of protection for influenza. And you can see here, depending on your baseline HAI responses, if it's low, it was much easier. 75% of these participants were infected, whereas a high baseline HI antibody, less than 10% of these participants could be infected. And this is an aspect that makes influenza studies much more challenging than cholera, for example, where we can assume everyone in the US is seronegative. We're going to have a variety of baseline immunity in these studies, and so to get that consistency of attack rate may be more difficult. Next slide. Now, this was a review that was done looking at randomized controlled trials for influenza drugs and vaccines. They looked at 26 published influenza challenge studies that were assessing interventions. Most of these assessed antivirals, but many did assess influenza vaccines, and actually a lot of live attenuated influenza vaccines were tested this way. Again, to emphasize, all the subjects were healthy adults between 18 and 65 years of age. Next slide. And likewise, in the US specifically, between the 1970s and 1990s, challenge studies were used for a variety of reasons, and again, were very critical tools to advance the influenza antivirals. Next slide. What happened, however, is there was an adverse event in 2000 that led to halting of US influenza challenge studies. And I've given you a lot of details on this slide, but I will summarize it by saying that this was a 21-year-old healthy man who had an adverse cardiac event. He did have some baseline abnormalities on his EKG before the challenge. He also had a high creatinine kinase, and these were attributed to exercise. Post-challenge, he actually did fine. He left the country for a few weeks. He had an upper respiratory infection and then came back on day 51 post-challenge and had a follow-up echocardiogram, which showed left ventricular enlargement and some severe systolic dysfunction. And it was never established what the cause was, and it was actually deemed unlikely to be due to the challenge virus. However, this event was concerning enough that in a risk-averse situation, again, challenge studies in the US came to a halt, although they did continue in Europe and elsewhere. Next slide. And it was really Matt Memely and the Laboratory of Infectious Diseases at NIAID that re-established and revalidated a challenge model beginning in 2012 and published here their dose ranging study in 2015 for an influenza A H1N1 challenge study. And you can see here that as we go up in dose, we have more symptoms, we have more viral shedding, and we have more participants who reach this endpoint of mild or moderate influenza disease. Next slide. And Matt Memley and his colleagues have used this challenge study in the last 8 years in a variety of ways. They have again validated the model to show that here, if you infect at time 0, we see an increase in viral shedding. We see symptoms follow 1 to 2 days later, and we see how they track over about 10 to 11 days. And that really informs how long we keep people in isolation. Next slide. And Matt and his colleagues have also used this to look at correlates of protection. And here again, you see some of the complexity of influenza challenge studies versus other challenge studies that we perform, in that the higher your baseline HAI considered as a single feature, the less likely you are to get influenza illness. The higher your neuraminidase titer at baseline, the less likely you are to get influenza illness. And the higher your stock antibody titer, the less likely you are to get influenza illness. So all of these at baseline correlate in some way with our ability to infect the participants. So you can start to understand how complex this baseline immunity is. And even if I can screen for somebody with a low HAI or a low neuraminidase or a low stock, there are other factors that might influence my attack rate for that particular virus. Next slide. And just to show here that, again, Dr. Memley and his colleagues have now established and validated a model for an H3N2 virus. Next slide. So how might we use this when we're looking at universal influenza vaccines? And this is just the target product profile that the NIH has put forward. Next slide. And we understand that these controlled human infection models help you in a number of ways. They allow you to reduce the variability of influenza. We know when we do field trials for influenza, we have an uncertain timing of the season. We can't predict the circulating strain. And in these challenge models, we can collect samples before and after infection. I've already mentioned that all adults are going to have some degree of previous exposure that is going to complicate this. And again, limitations are that these are performed on healthy young adults, and that the mode of transmission and inoculum, we try to replicate what's happening in the real world, but we actually don't know, for example, the exact inoculum in a natural setting for influenza. Next slide. However, we do know that with these field trials, they're expensive, they're multi-site, multi-year. You know, we often don't get to assess durability of immunity. And particularly if we're looking at broadly active influenza, you rarely have an influenza vaccine trial where both influenza A strains and one or both influenza B strains are circulating. We also don't have the ability to know if we're looking at a matched strain or a heterologous strain. So that's a powerful use of a challenge study. Next slide. And again, just to show this example in our pivotal cholera challenge study, for both the late and delayed, when I combine those together, we had about 130 participants that gave us a highly statistically significant endpoint. For a typhoid challenge study, it took about 100 participants to show a highly statistically significant endpoint for vaccine efficacy. I just finished a field trial of typhoid vaccine in children. It took 30,000 children to show that statistically significant endpoint. So you see there the power of the smaller sample size on what we can do with these studies. Next slide. And this is just an example of an influenza study. You know, if we're looking at a 5% attack rate in the field. Next slide. And the difference between what you need for a field efficacy study at any point efficacy you would like to establish versus if you have a 70% attack rate in a challenge study and the number of participants that's needed. Next slide. So really just finishing up here, we actually completed a challenge study last year, and one of the aims here was to establish that an influenza virus controlled human infection model could be done in a standardized way at multiple sites. And so we went to 4 sites, Duke, St. Louis here, and Cincinnati. Because of COVID, we've had some delay in getting our results, so I don't have results to present to you, but I can tell you that there was consistency of attack rate among the sites. And because we have to bring these participants into challenge units, because we have to keep them in isolation for a period of time, it's really going to take standardized multi-site studies to be able to assess vaccine interventions. Next slide. So Ted mentioned the civics, and Duke and the University of Maryland, where I am, we are the 2 clinical cores, and one of our objectives will be to look at vaccines using healthy volunteer challenge studies. Next slide. So to wrap this up, these human challenge studies have been successfully employed with a number of different organisms to understand pathogenesis, immune responses, correlates of protection. They can be done at lower cost than pivotal field trials, and they may help justify further investment on product development, even if you intend to ultimately do a field trial. And we know in the US, for example, it's very hard to do a field trial with a placebo control group anymore. So these are huge investments to take influenza vaccine all the way to licensure. We do know, though, that it won't be easy with influenza. We have underlying immunity, we have generalizability, we have to manufacture strains. If we're going to look at heterologous strains, we have to decide what strains to manufacture. It takes time to manufacture strains, and while we're manufacturing them, the population is developing immunity. So it's really quite complicated. But again, could be a very powerful tool to look at breadth and duration of protection and establish correlates. So thank you very much. I'd be happy if there's time to answer questions.
Thank you, Kathy. That was excellent. We do have a question in the chat, which I'm going to read to you from Dr. Vogel. She's asking if, how big are the challenge studies typically? And are there implications with respect to the interpretation of the results, lower statistical power, or is it outweighed by the clearer signal in the results as the challenge studies are more controlled? I know you kind of addressed some of that.
Yeah, so I think the sample sizes can vary, and as I showed in that one slide, it's really going to depend on what your goal is. If you're looking at vaccine efficacy, I think the biggest unknown in influenza challenge studies is the attack rate. Because we can look at a malaria challenge model, and we can look at a cholera or a Shigella, and you're starting with seronegative participants. We can understand the inoculum that will give us predictably a 60 to 70% attack rate. That is much harder to do with influenza. And with influenza, you have to reestablish that with every new challenge strain. And so that's where it really becomes difficult. If you can reach that 70% attack rate, as I showed, you can get very powerful results with 20 to 80 people. It doesn't take a lot of people, but it's just getting that consistency of attack rate and that generalizability of participants.
Right. And it sounds like both, field trials and the challenge strain kind of trials both give you powerful sets of data. And it really depends on the questions you're asking and whether you have the finances to go to a field trial or go straight. But they both have their pluses and minuses, it seems, and challenges.
Yes, absolutely.
Well, thank you very much, Kathy. Greatly appreciate your presentation.